Hi everyone, am I audible? Good morning. Am I audible? Good morning everyone. Am I audible and visible to you all? Good morning. Welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma, and today we are going to continue sexual reproduction in flowering plants. So basically I think from near about 10 days we were missing on YouTube, है ना? We didn't take any live sessions. So from today, I'm going to start it and we'll finish the sexual reproduction in flowering plants. And in the evening marathon, we will discuss breathing and exchange of gases. We have already completed one chapter of human physiology, that is digestion and absorption. We have even completed the MCQs, top MCQs of digestion and absorption. And now we'll finish in this particular week, we will finish sexual reproduction in flowering plants and breathing and exchange of gases as well. Okay. So soon I will increase the duration of my sessions as well, and even the frequency as well. Okay. So before the summer, we'll try to finish our syllabus. Then there will be the detailed marathons, one shots, detailed marathons, so we are already taking, but there will be the one shot, there will be the quick revision sessions then. Okay. So thank you so much. So do you know why do we celebrate Teachers' Day? Yes, I hope you know about it. Why do we celebrate Teachers' Day? Do let me know after the class in the comment section. So now let's start the class. Let's continue with this particular chapter, that is sexual reproduction in flowering plants. And yes, it is one of the easiest chapters. So you can expect 3 to 4 questions from this particular chapter, right, students? And moreover, I need to show you something. See this video. This particular video here, I have shared the strategy with you all, right? The complete roadmap to score 650 plus marks in 239 days. Actually, see, if I talk about your NEET examination, so we can expect your NEET examination in the first week of May and the first Sunday, that is your 5th of May, right? So as per that, I have made this strategy video, and here I have divided, right, each and every day equally. I told you, I have already explained in this video that how many days are left, how are you going to utilize it, right? The duration for the revision is also included there, so do check out this video. Okay. And moreover, students, here you guys can see the link for our batch, right? In Avengers 2.0, currently I'm teaching human physiology. If you enroll in this particular batch, right, obviously you will get this live classes. 70% of the syllabus will be live and 30% will be in the recorded form, right, that we have already completed. And after every 14 days you will get the mock test as well. Okay, so you are completing the syllabus as well and you are practicing the questions as well. So kindly join this batch, because soon we are going to close the enrollment for it. And trust me, right, this batch is near about 5,000 rupees, right? This is what you have to pay for this particular batch. Okay, and here you are going to get the quality content from the experienced team. We know what to deliver, we know what is important, what is not. So accordingly we are going to prepare you, right? And moreover, doubt sessions will be provided to you, discussion sessions will be provided to you. So this batch is as per the students' need. If you need any extra classes in a particular topic, we are going to provide you that. If you need the discussion on a particular topic, we will schedule the classes accordingly. So be the part of this batch as soon as possible, because soon we are going to close the entries for this batch as well. Okay, and see, if you will join it late, it will be of no use, right? Because then there will be so many classes that you need to cover. Okay, so please join it now. Still you have sufficient time, and we have designed the schedule in this way that you will get the time to cover the backlogs as well. So join it as soon as possible. Okay, now come back to this topic, and you know that already what we have covered. So we have started with the yes, so you know that we started with the flower. So flower is the site of, or it is the seat of sexual reproduction, right? It is the reproductive organ, that is flower. And related to flower, we have already discussed the 4 whorls, the essential whorls, the non-essential whorls, even the male part, even the female part. So today let's revise the female part first, and then we will start the discussion on the ovule. So tell me, in the case of flower, which one is the female part? Yes, tell me, in the case of flower, which one is the female part? Gautam bache, join our Telegram channel bache, and you will get the schedule there. Yes, which one is the female part when we talk about the flower? Which one is the female part when we talk about the flower? Of course, you are right. It is gynoecium. What is it, students? It is gynoecium, that is the female part, right? So when you talk about this flower, which is the reproductive organ, which is having the 4 whorls, this gynoecium, which is the innermost whorl, it is the female part. And this gynoecium, it includes what? It includes pistil or the carpel. Okay, it includes what? It includes the pistil or the carpel. So here we can see that there can be single carpel also. Then the flower will be monocarpellary, and there can be many carpels also, right? Then the flower will be your multicarpellary. Okay, the flower is going to be multicarpellary then. Okay, so monocarpellary, multicarpellary. So mono means single, multi means many carpels are there. So accordingly, you know that in the flower we have the arrangement as well. Okay, what we have? We have the arrangement as well. Let me start this part. Okay, so if the flower is multicarpellary, we have completed it in last class. Let's revise it quickly, right? If so many carpels are there, right? If so many carpels are there, so you know that there will be 2 fates, isn't it? Like, let's say in a family, if there are so many members living there in a family, either they are living very lovingly, right? Or they have that, you know, fights and all, right? Right, you can relate it with the joint families. So when the flower is multicarpellary, we have 2 scenarios. One is apocarpus, another is syncarpus. What can be there? We'll see apocarpus flower, we'll see syncarpus flower. So A for alone. This is what you need to remember, right? A for alone. And here syn means fused. So if all that carpels, right, they are present in a flower but they are not fused together, they are living singly, then it will be the apocarpus condition. Okay, we have so many carpels in a flower and all that carpels are living individually, they are living singly, they are not fused together, right? See, when you talk about the carpel, you know that what do we have? We have stigma, style and the ovary, isn't it? We have stigma, style and the ovary, isn't it? So all that carpels, they are living individually, right? So it is the apocarpus condition. So I have given you the trick for it. The trick is PAM. What is the trick for this apocarpus flower? It is PAM. So can you tell me, bache, what is the meaning of this PAM here? Yes, everyone, can you just tell me what is the meaning of this PAM here? Yes, if you are a regular student, you know about it. Very good, Priyanka. Just tell me what is the meaning of the PAM here. I'm talking about the examples, and examples are important, right? Examples are important. Tell me what is the meaning of this PAM here, or it should be tell me. Pam or ma'am? What should be there? Very good. It should be ma'am. It should be ma'am. So 1M means multicarpellary, right? 1M means multicarpellary. A means apocarpus. A means apocarpus. And then M means mycelia. So mycelia is the example of the flower, right, where you will see apocarpus condition. So mycelia is given in your class 12th NCERT, but other than mycelia, even the rose is there, even the lotus is there, and it is given in your class 11th NCERT. So we have to revise both. Okay, so rose, lotus and mycelia, they are the examples of the apocarpus flower, right, bache? Rose, lotus and mycelia, they are the example of the apocarpus flower. Now when it is syncarpus, the trick is going to be PMS. What is the trick here? It is PMS. Again, P stands for paperwort, M stands for multicarpillary, right? And S stands for syncarpus. So students, examples are important here. Okay, examples are important here. Clear, bache? Clear, bache? So other than papaver, you can also note down one another example, that is your hibiscus. Hibiscus, you know na, China rose. China rose is also having syncarpus condition. Clear? The China rose is also having the syncarpus condition. Clear, bache? Clear, bache? So this stigma, style and ovary, it is making that carpel and the pistil. Now we have to talk about this part, right, bache? So you know that ovary is the swollen part, right? It is the swollen basal part, and the space here within the ovary, it is known as ovarian locule, right? What is it? It is known as ovarian locule, or you can call it as, simply you can call it as locule or the ovarian cavity. Right, this is the word that you can use. So what do we have here? It is the locule, or you can also use the word cavity, ovarian cavity. Are you getting my point? Right? Are you getting my point? So, bache, here in the ovary, we can see bilocular, we have the bilocular ovaries as well. Multilocular ovaries are also there. Trilocular ovaries are also there. So, if there is only one locule, then it will be unilocular. If two locules are there, bilocular. If three locules are there, trilocular. If there are so many locules, then it will be the then obviously multilocular ovary it is. Clear, bache? Like we see in the case of exile placentation. I hope you remember the exile placentation. Do you remember exile placentation? We see in the case of lemon, in the case of your tomato, right? Basically in the case of citrus fruits, we see the exile placentation. So you see here you have so many seeds, isn't it? You see so many seeds. So actually, bache, ovule after fertilization is going to form the seeds. Okay, ovule after fertilization, post fertilization, from the ovule we will get the seed. So if in a particular fruit you see so many seeds, it means it is having so many ovules. Clear, bache? And here, like take the example of the lemon. So this part is the locule here, right? This part is the locule here, and the seed is showing what? It is showing the ovule. So after fertilization, ovule will form seed, ovary will form fruit. Any doubt here? Yes. Any doubt here? Right? Because today we need to discuss the structure of ovule, but before that, this is what you need to understand. And mostly students, mostly in one locule, we used to see one ovule. Okay? In one locule, yes, Meenakshi, in one locule, we used to see one ovule. Clear? Yes? Is it clear? Done? So now let's start the structure of the ovule. So see, this part is the ovary part, है ना? This part is the ovary part. So here in this ovary, this ovule, let's see, this is how the ovule is present. But actually, na, this ovarian wall, near to this ovarian wall, we have the placenta here in the flower. What do we have here, students? We have the placenta here. What do we have here? Yes, so Hashini, what do we have here? We have the placenta here, and you know that this placenta is important for the nourishment. Okay, so this ovule, it is attached to the ovarian wall, it is attached to this placenta. Actually, from this placenta there will be a stalk-like structure that will arise. Okay, there will be a stalk-like structure that will arise, and this stalk-like structure, which is attaching this ovule to this ovarian wall, which is attaching this ovule to this placenta, it is known as your funicle. What is it? It is known as your funicle, right? Right? Let's revise it again. What are we discussing now? We are going to discuss the ovule. What is this ovule? Ovule is the megasporangium. What is it? It is the megasporangium. It is the structure in which megaspore will form. Okay, it is the structure in which megaspore will form. So where are we going to get this ovule? In the ovarian cavity, in this locule. Clear, bache? So here I'm saying that this ovule is attached to the ovarian wall, right? This ovarian wall is having the placenta here. Okay, this pink colored structure that I'm showing here, it is placenta. Now we have to attach this ovule to this placenta. And where is this placenta present? This placenta is present near to this ovarian wall. This placenta is here, right? This placenta is here near to this ovarian wall, right? So a stalk-like structure is attaching this ovule to the ovarian wall, and that stalk-like structure is known as, yes everyone, that stalk-like structure is known as funicle. What is it? Yes everyone, what is it? It is funicle. So ovule is attached to placenta by the means of this funicle, right? Ovule is attached to the placenta with the help of, or with the means of, this funicle. So it is that stalk-like structure. Okay, it is that stalk-like structure. So now this is the structure that we are understanding. Okay, so right now to this, so I will I will magnify the structure, right? And then I will show you each and every part here. So first thing that you need to remember is that this is the ovarian wall. It is having placenta which will nourish this ovule, and this ovule is attached to this placenta with the help of stalk-like structure that is known as funicle. Okay, that is known as funicle. Now bache, let's say this is the funicle. Okay, and here you have the ovule. So the point where this funicle is touching the ovule, it's very simple and easy. The point where this funicle is touching the ovule, it is known as hilum. What is it? It is known as hilum. You can also call it like this. The point where funicle is fusing with the ovule, it is known as hilum, or you can simply say the junction of, right? Or you can simply say junction of this junction where funicle is attaching with the ovule, it is known as hilum. Clear bache? It is known as hilum. What is it? Yes everyone, what is it? Tell me, what is it? It is hilum. So hilum represents the junction of funicle and ovule, right? What is this hilum representing? It is representing the junction of funicle and ovule. So I'm explaining it again. This funicle, it is getting attached to this particular ovule, bache, right? So this point where this funicle is fused with the ovule body, it is the hilum, or in another way you can say that hilum is the junction between this funicle and ovule, right? It is the junction between this funicle and the ovule. This is what hilum is. Clear bache? This is what hilum is. Done? Yes, is it clear? Is it clear? Funicle clear and the hilum clear. So with the help of this funicle is having the vascular strands. Okay, this funicle is having the vascular strand, your xylem and phloem, and it is providing nourishment to this, right bache? It is providing nourishment to this particular ovule. Is that clear? It is providing nourishment to this particular ovule. Now this major part of this ovule, this portion here, right? This portion here, bache, it is the parenchymatous portion. It is your nucellus. What is it? It is the nucellus. Yes, what is it? This particular portion here, Srilekha, like this one. This is what? This is nucellus, and nucellus is the living portion. It is the parenchymatous portion. But we have already covered the anatomy of the flowering plant. So I hope you are aware of this parenchyma. Parenchyma is the living tissue having, thin-walled cells, right? Remember, having thin-walled cell. So this majority, right? This major portion, right, majority of the part of ovule, or this major portion of ovule is what? It is the nucellus, which is parenchymatous, which is living, and it is having the reserved food. Clear bache? It is having the reserved food. So first of all, you need to understand the structure of megasporangium, because if you will understand the structure of megasporangium, you will understand the structure of the embryo sac, which will develop here. Clear bache? Clear bache? So what is ovule? Ovule is the megasporangium, the structure in which megaspore will form. Okay, so this stigma style ovary, this carpel, it is equivalent to megasporophyll, and this ovule is what? It is the megasporangium. Clear bache? This ovule is what? It is the megasporangium. Are you getting it? Are you getting it? So that's how you have to remember. Clear bache? Clear bache? That's how you have to remember. Done? So now this nucellus is covered with the coverings. Okay, this nucellus, it is covered with the coverings, and these coverings are what? They are known as, yes bache, they are known as integuments. What is it? It is the integument. So this nucellus is covered. It is having the covering. The name for that coverings is integument. What is it? It is integument. So in general, integument means covering. So here in the case of this ovule, you will see that we have actually it is a bitegmic ovule. What is it? It's a bitegmic ovule. What is the meaning of bitegmic ovule? Bi means two. What is the meaning of bi, Priyanka? Yes, bache, Suhashini, Mohammed. What is the meaning of bi here? Bi means two. Tegmic, bitegmic ovule means it is having two integument, right? So this one is your inner integument, right? And this one is your outer integument. Clear, bache? So two outer coverings are there. Outer integument, inner integument. Outer integument, inner integument. And these integuments, after fertilization, they are going to form the seed coat. Clear, bache? These integument after fertilization, okay, they are the one which will form what? They are going to form the, yes, they are going to form the seed coats. Like your outer integument will form testa, your inner integument will form tegumen. Clear, bache? Clear, bache? So here we have the example of bitegmic ovule. Other than that, we are also having unitegmic ovule as well, in which only one, right? In which only one covering is there. Are you getting it? Are you getting it? Here we have the example of ovule where two integuments are present, but we also have unitegmic ovule that you will see in the case of gymnosperms. Where will you see it, Suhashini? You will see it in the case of most of the gymnosperms. They have unitegmic ovule as well, and even in the case of angiosperms, in few angiosperms you will see unitegmic ovule. Clear? Now this integument, it is not covering the entire nucellus. See, at the tip this integument is missing, and this end we will call it as micropyle or the micropylar end. What is it? It is the micropyle or the micropylar end, right? So I'm repeating this portion again. This is the ovary part. Ovarian walls are having the placenta to right. Now here we have the ovule. We need to attach this ovule to placenta. Why? So that it can get the nourishment. So the funicle is going to attach the ovule to this placenta, and this funicle is having the vascular strands which are going to provide nourishment to what? To the ovule. Now the junction at which funicle touches the ovule, we used to call it as hilum. What we used to call it? We used to call it as hilum, right, bache? Right, bache? And the major part of this ovule here is the nucellus. It is the parenchymatous part. It is having the reserve food material, and it is the portion where we will see the embryo sac will form. Clear, bache? Clear? Now this nucellus is covered with the coverings. We used to call it as integuments. We have outer integument also. We have inner integument also. So if we have two integuments, such ovule is bitegmic ovule, right, bache? If there will be single integument, that ovule will be the unitegmic ovule. So these integuments are not covering the entire nucellus. See, at the tip integuments are missing. So this end is the micropylar end, and you can simply call it as micropyle. Okay, you can simply call it as micropyle. Clear, bache? Clear, bache? So opposite to this micropyle, always remember, opposite end to this micropyle, it is known as chalazal end. Right? Opposite end of micropyle, it is known as chalazal end. What is it, students? It is known as chalazal end. Are you getting it? Sure? Sure, always. So now if you look at this particular ovule, you will see that hilum, chalazal end, micropyle, they all are in a straight line. Yes or no? Can you see that? Can you see that? That micropyle, your chalazal end, your hilum, they all are in a straight line. Right? They all are in a straight line. So such type of ovule is your upright ovule. Right? Such type of ovule is your upright ovule. Why? Because your micropylar end, chalazal end, hilum, funicle, they all are in a straight line. Right? They all are in a straight line, and it is also known as your atropous, right, or orthotropous ovule. Clear, bache? It is known as atropous or the orthotropous ovule or the upright ovule. Clear, bache? Clear, bache? So it is known as atropous, orthotropous orthotropous or the upright ovule. Clear? Clear, bache? Right, but in your NCERT, we talk about the inverted ovule, right? What we discuss? Anatropous ovule. So if you want to understand the structure of anatropous ovule, you should know the simple ovule, right? You should understand the simple ovule. So this is what you need to keep in your mind, that wherever funicle is touching ovule, the junction is hilum. Clear? Opposite to micropylar end, there will be the chalazal end. If these points are clear to you, then you can easily draw anatropous ovule. Clear, bache? You can easily draw anatropous ovule. So basically there are different type of ovules, but in your syllabus, in your NCERT, we talk about the anatropous ovule only, the inverted ovule. It is inverted at 180 degree, and why? Right, in most of the angiospermic families, actually you see that anatropous ovule, right, bache? You see that anatropous ovule. Clear, bache? Clear. So that's what you need to remember. Okay, that's what you need to remember. So I'll add few more points here. Now see, if your ovule is inverted, let me draw that. Now see, it is something like this. Okay, done. Let's say it is something like this. It is inverted, right? It is the inverted ovule. Anatropous it is. Now see, again the diagram is easy. Now let's say if this is the funicle and this is the point where it is touching the ovule. So obviously this part will be the, right, this part will be your hilum. Like let's say if this is the funicle, here it is touching the ovule. So don't you think that this part will be the hilum? Yes or no? That this part will be the hilum, yes or no? I told you, the structure is easy. This stalk-like structure is going to be your funicle. And see, this is the part where integuments are not covering the ovule. So this will be the micropylar end. This will be the micropylar end, and opposite to micropylar end, you know what you have? You have the chalazal end. So here you can see that micropylar end, chalazal end, hilum, funicle, they all are not in a straight line, right, bache? They all are not in a straight line. So this is your inverted ovule. This is your anatropous ovule. What is it? It is your anatropous ovule. Is that clear? Yes, everyone. Is that clear? Yes. Done. So sometimes, bache, what is the scenario that this funicle, it touches the ovule, right? Right. No doubt it touches the ovule. Like towards the lateral side, it is extended a little. Okay. Sometimes this funicle, when it touches this ovule, right, towards the lateral side, it extends a little. Then it forms some outgrowths, and we used to call it as raphe. What we used to call it? We used to call it as raphe. So what is a raphe? They are some ridge-like structures, right, present on the lateral side. Okay, bache? So funicle, when it is attached to the main body of the ovule, for some distance at lateral side, it forms a ridge-like structure. For some distance at lateral side, it is forming what? It is forming a ridge-like structure. And what we used to call it? We used to call it as raphe. What we used to call it? We used to call it as raphe. And other than that, we have also aerial. Do you know aerial? Aerial is known as third integument. Right? This is important. Aerial is also there. Aerial is known as third integument. So you will find aerial in the case of lychee. Do you know what is aerial? Anyone? Anyone here in class? What is aerial? Yes. Do you know what is aerial? We consider it as a third integument. Yeah, it is there in the lychee. So actually, bache, it develops from funicle at the base of ovule. Okay, it develops from funicle at the base of ovule. Clear? At the base of ovule. So this is about the structure of ovule, which will form your future seed. Clear? So if there is any doubt, do let me know. Quick. If there is any doubt, do let me know. Yes? Sure? Are you sure? Thank you, bache. Okay, so this is the structure of ovule. What is it? It is the structure of ovule, and this is what you need to remember. Okay, so anatropous ovule is given in our syllabus. Okay. Okay, so now we have to discuss the development of the ovule. Okay, and then we'll be talking about the megasporogenesis. Fine. So first of all, let's talk about the development of ovule. And students, this chapter is very easy, right? In the form of a story, this is what you need to remember. And then you need to focus on the examples mainly, that which one is the apocarpus flower, which one is the syncarpus flower, and the examples in the case of pollination. Okay, so this is the part that you need to remember. Otherwise, this chapter is the easiest chapter, right? If you will understand it, right, if you will cover it in this particular lecture, then later on you just need to read it from NCERT, that's all. Okay, NCERT is more than sufficient for this particular chapter, and after that just practice the previous year questions. And every time you can check any of the paper, right, you will find same kind of questions from the sexual reproduction and flowering plants. So from sexual reproduction and flowering plants, from biotechnology, right, you will get almost same type of questions you are going to get. So if you have solved the previous year questions, then there is nothing to worry about. Paper is going to come from that part only. Okay, your paper is going to come from that particular part only. Done, bache? So next portion that we need to study, as I said, it is the development of ovule. What is it? It is the development of ovule. Ovule is what? It is the megasporangium. Now, this is the ovary wall, and as I said, that this ovary wall is having the placenta. So actually, bache, when you talk about the development of the ovule, from this placenta, you know that from this placenta, your nucellus form. I'm telling you the initial stages of development, then slowly we will discuss megasporogenesis and development of female gametophyte. Clear? So when we talk about this placenta, right, this placenta is actually forming the nucellus. Okay, this placenta is actually forming the nucellus. So initially, during the development of this ovule, this nucellus is nothing, it is just the mass of some undifferentiated cells. You know this process, right? We have completed the anatomy. Okay, we have completed the anatomy. So initially, this nucellus is what? This nucellus is what? It is just the mass of undifferentiated cell. So, bache, if I'm using the word undifferentiated cells, so can I say that they are meristematic here? Can I say that they are meristematic here? If I'm saying that they are undifferentiated, means that they do not have a proper fate, right? They are just dividing. They do not have a specific function, isn't it? Isn't it? They do not have a specific function. They are just meristematic. They are just meristematic, and they are dividing and dividing. Yes or no? They are just dividing and dividing. They are meristematic. Clear, bache? Clear, bache? So with time, these undifferentiated cells, they will become parenchymatous. Right, they will become parenchymatous. So just imagine this process. Imagine this process. Your flower is developing. The ovule is developing. So from that placenta, from the cells of that placenta, the cells of placenta, they will form a mass of cells. Right, and that mass of undifferentiated cell, it is actually the nucellus. It will divide, and then it will acquire a permanent function. It will form this they will become parenchymatous. Clear, bache? They will become parenchymatous, right? They will become parenchymatous. So actually, right, so as of now, I'm just focusing here on this nucellus, right? Otherwise, you know the entire structure, that nucellus, it is covered with the integuments, right, bache? Here you have the funicle and all. You know this complete structure. But as of now, what am I teaching? Right, I'm teaching you about the nucellus, right? So now what is going to happen here in this nucellus? It is also interesting. So here in nucellus, which is a mass of parenchymatous cells, see, this outermost layer is the layer of epidermal cells. Yes, here also in the case of nucellus, we consider it like this. This outermost layer is considered as the layer of epidermal cells, or you can simply call it as epidermal cells. So below this epidermis, obviously, there will be hypodermis. These terms are simple. Below the epidermis, what do we have? We have the hypodermis. Now, bache, in this hypodermal region, near to the micropylar end, right? Near to the micropylar end, let's say, you are talking about this particular cell. Ultimately, it is the mass of living cell. Ultimately, all the parenchymatous cells are there. So near to this micropylar end, somewhere here, right in this hypodermal region of nucellus, or simply you can say that in the nucellus, near to the micropylar end, we have a particular cell here. Now this cell is actually your archisporial cell. This is the cell which is going to form your megaspore mother cell. Okay, so why this cell is archisporial cell? Because this cell is having a distinct nucleus. Okay, it is having the distinct nucleus. Its size is larger than other cells. Okay, so this is a kind of cell which is having the potential to make the megaspore mother cell. Okay, so near to this micropylar region, here in this hypodermal cell, you have a particular, right here in this hypodermal region, you have a particular cell, and we are calling it as archisporial cell. Which cell it is, Subhashini? It is the archisporial cell. Which cell it is? Yes, everyone? Which cell? Which cell is there? It is the archisporial cell. Let me draw it once again. Okay. So now we will talk about the archisporial cell only. Okay. So you know that this one is what? This one is your nucellus. So, or take it like this. For your better understanding, consider it like this. Clear? Take it like this. So now this archisporial cell, of course it is diploid. It is having a distinct nucleus, having the dense cytoplasm. Now it will divide. It will undergo division. Okay, what is going to happen here? Just a minute. But just for your understanding, I'm drawing the structure. Ultimately, this is what you need to understand, that this process is going on within the nucellus. Okay, so this is the archisporial cell. As I said, this archisporial cell will divide. Okay, it is dividing mitotically. You will get two cells, right? One is your primary parietal cell, another one is your primary sporogenous cell. Clear, bache? Clear, bache? So archisporial cell, it will divide mitotically. It will form primary parietal cell, primary sporogenous cell. Clear, bache? Primary parietal cell, primary sporogenous cell, right? So how is it dividing? It is dividing mitotically. So if mitotically it is dividing, so obviously these two daughter cells are also diploid. These two daughter cells are also diploid. So the cell which is near to the micropylar end, this is actually the primary sporogenous cell. What is it? It is the primary sporogenous cell. And this one will be your primary parietal cell, right? This one is what? This one is your primary parietal cell, right? So let's just focus on this primary sporogenous cell here. So it is sporogenous. What is the meaning of sporogenous? It is the one which will form the spore. It is the one which will form the spore. So actually this primary sporogenous cell, it will start behaving like your megaspore mother cell. Okay, so here we have the ovule. This part is the nucellus. Just below the epidermis, you have the hypodermis here. Here you will see this archisporial cell. This archisporial cell is diploid. It is mitotically, it will divide. It will form primary sporogenous cell, primary parietal cell, and this sporogenous cell will behave like the megaspore mother cell. Right? It will behave like the megaspore mother cell. Right, bache? Right, bache? It will behave like what? It will behave like what? It will behave like megaspore mother cell. Megaspore mother cell. Is that clear? Is that clear? Now you know, see, MMC, that is megaspore mother cell. So I told you before that if it is a mother cell, definitely it will undergo meiosis. And if I talk about the ploidy of this megaspore mother cell, 110% it is diploid, right? 110% it is diploid. So let's say if this one is your diploid megaspore mother cell, so of course it will undergo meiosis 1 first, which is the reductional division, right, bache? Which is the reductional division. Clear, bache? Clear, bache? So you will get 2 haploid megaspores first, and then there will be the meiosis 2, which will result in the formation of 4 haploid megaspores. Clear? Which will result in the formation of 4 haploid megaspore. So this diploid megaspore mother cell will undergo meiosis 1, and here you will be getting 2 haploid megaspores, and then there will be the meiosis 2, and you will be getting 4 haploid megaspores. Clear bache? Clear bache? Yes. So where this process is taking place? It is taking place within the nucellus. Okay, okay, because spore formation is there in the ovule, right? So that's why we used to call it as megasporangium, because spore formation is there in the ovule. That's why we are going to call it as megasporangium. Are you getting my point? Right, that's why we are calling it as what? We are calling it as megasporangium. So now is it clear? Now is it clear? But now here we have one more point that we need to understand. You know that in the nature, female gametes are comparatively less, right? Male gametes are more in number, female gametes are less. Why? Because it is the female gamete which will grow, which will after fertilization, which will form the zygote. It is the one which will, right, which will further develop and will give rise to a new life. Like in the case of humans also, you know that during the sexual intercourse, male is going to ejaculate so many sperms, right? So many sperms in the female vagina, but in the female vagina there will be only one female gamete, right? Right? So one sperm will fuse with that one gamete only, and then it will grow and it will form the zygote, right? Which will get implanted, and you know that the further process of that pregnancy, isn't it? Isn't it? And here also, because it is the megaspore is the one which will form your female gamete, right? So here also we will see, right, that female gametes are less in number, male gametes are more in number. So here what is going to happen? No doubt, megaspore mother cell will form 4 haploid megaspores, but out of it only 1 is functional, 3 will be degenerated. Okay, only 1 will be functional, 3 megaspores by an unknown mechanism, 3 megaspores which are present near to the, right, which are present near to the micropilar end, they will degenerate, and this only 1 megaspore will be the functional one. Okay, only 1 megaspore will be the functional one. Are you getting it? It will be the functional one. Are you getting it? Right? The megaspore near to the chalazial end. So this one will be the functional one. So it will grow and form embryo sac, and rest 3 they will degenerate. So if in the paper there is a question that 200 megaspore mother cells, how many megaspores will be formed from 200 megaspore mother cell? What will you say? How many megaspores will be formed from 200 megaspore mother cell? What will you say? But let me explain this once again. This is the nucellus. Here you have a cell. This is the archisporial cell. This archisporial cell is having a very distinct nucleus. Because of it, it is growing in size and it is dividing mitotically. So when it is dividing mitotically, it will form primary sporogenous cell and primary parietal cell. The cell which is near to micropilar end, it is your sporogenous cell, the one which will form the spore. Which spore? Megaspore mother cell. Okay, megaspore mother cell, right? And rest this parietal cell will divide and it will form, you know, other part of this nucellus actually, right? This is the initial developmental stages, and accordingly I'm telling you. So this megaspore mother cell, it is diploid, and when it is the megaspore mother cell, 110% it will undergo meiosis, right? Which is a reductional division, right? It will undergo meiosis, which is a reductional division. So firstly, it will undergo meiosis 1. It will form 2 haploid. It is going to form 2 haploid megaspores. Then there will be the meiosis 2, which will form 4 haploid megaspores. But in the nature, female gametes are less. So out of these 4 megaspores, only 1, which is near to chalazial end, it will remain functional, and rest 3, they'll be degenerated. Okay, they will be degenerated. Clear, bache? They will be degenerated. Only this one will be the functional one. Now clear? Now clear? So I asked you one question that 200 megaspore mother cell, they will form how many megaspores? Yes, they will form how many megaspores? That's what I'm asking. I've already repeated it, bache. Now you answer my question, please. And if you're new to our channel, you know what you have to do. Just subscribe our channel. Just subscribe our channel. Yes, my question is that if you have 200 megaspore mother cell, then they will form how many megaspores? Then they will form how many megaspores? That's my question. That's my question. Please answer. Please answer. Bache, no. My question is how many megaspores will form? So from 200 megaspore, if one is forming 4, so 200 into 4, obviously it will be 800 megaspores. 800 megaspores. But now my question is, now listen to my question once again. How many functional megaspores will be formed from 200 megaspore mother cells? Right? Very good, smiling. Excellent, bache. Right, initially I asked you that how many megaspores will be formed from 200 megaspore mother cell. So obviously if one megaspore mother cell is forming 4 megaspores, so 200 will form 800 megaspores, right? But out of it, how many are functional? Now I'm saying how many are functional. So obviously 200 functional megaspores will be formed. Clear, bache? 200 functional megaspores will be formed. So now this megaspore, okay, now this megaspore, it will form the embryo sac. And this process of formation, right, this process of formation of megaspore from megaspore mother cell, it is known as megasporogenesis, right? The process of formation of megaspore mother cell, megaspore from megaspore mother cell is the megasporogenesis. Is that clear? Is that clear? It's very easy now. It's very easy. What is megasporogenesis? Megasporogenesis. Genesis means formation. So formation of megaspore. So it will be megasporogenesis. Formation of megaspore. It will be megasporogenesis. That's what you need to keep in your mind. Clear, bache? Clear, bache? So if I compare it with the male part. So in the males, if you remember, right, that tape it up. Remember? Wait. Let me show you the diagram. See, in the case of male part, you know that here, right, this tapetum is covering the sporogenous tissue. Here, each and every cell is having the potential to form the microspores. Remember in the case of males. So here you will see so many microspores. Okay, here you will see that one microspore mother cell or pollen mother cell is forming 4 microspores, and these microspores will further grow into the pollen grains, right? They will further grow into the pollen grain. So here we will, what we have studied, that one pollen mother cell, it will form 4, right? 4 microspores, and they all are functional. Clear, bache? They all are functional. But here only in the case of females, only one is functional. Done, bache? Only one is functional. So now that's what we need to study. That is development of your female gametophyte. Clear, bache? What we need to study now? We need to study the development of your female gametophyte. That is embryo sac. Clear? So any doubt? Yes? Navash, which will not reductional? I didn't get your question. I didn't get your question. Okay, so from this part we are going to start here. See, so near to the chalazal end, here, near to the chalazal end, you have one megaspore. Near to the chalazal end, you have one megaspore which is functional. You have one megaspore which is functional. Remaining 3 are degenerated. Remaining 3 are degenerated. Is that clear? Is that clear? Clear, bache? So now this single, right, now the single megaspore will develop. Wait. Now, this megaspore, it is haploid, bachu. This megaspore is haploid. Yes, this megaspore is haploid. Yes or no? So now this haploid megaspore, it will undergo mitosis. Mitosis is the equational division. Mitosis is the equational division. So what is going to happen here? It is nuclear division will take place. Now, immediately after the karyokinesis, there is no cytokinesis. What is going to happen here? This in this particular megaspore, what will happen? The nuclei will divide. So it is the mitosis, which is the equational division. So after first, right, there will be the mitotic division. So after this mitosis, here in this megaspore, now bache, this megaspore is developing. Okay, megaspore will form female gametophyte. What is that female gametophyte? Female gametophyte, the structure which will form female gamete, and that is your embryo sac. What is it? That is your embryo sac. Right, bache? Right, bache? So single spore, single functional megaspore is growing, it is developing and forming female gametophyte. Right? So such type of development where one single spore is forming female gametophyte, it is known as monosporic development. Right? It is known as monosporic development. The single spore is developing. Clear? It is the single spore which is developing. Clear, bache? Clear? So there will be the mitosis. So after mitosis, here you guys can see, right? Here you people can see, you will get 2 nuclei. Then again there will be the mitosis, right? Again there will be the another round of mitosis. Actually there will be 3 mitotic divisions will occur, right? One after another, 3 mitotic divisions will occur. So see, after first mitotic division there will be 2 nuclei. After second mitotic division there will be 4 nuclei, right? And after third mitotic division there will be 8 nuclei, right? There will be 8 nuclei. Clear, bache? So understand this complete process, right? Try to understand this complete process, right? Imagine this process. How did we start? We started from this particular part, right? We started from this particular part, that this is the carpel that we have here. This is the swollen part is the ovary. Ovary is having ovarian locule where your ovule is growing. Right? That's how we started. Let's revise it very quickly, students. Right? Let's revise it very quickly. So here to the ovarian wall, your placenta is attached and this placenta is going to form your nucellus. Clear, bache? So actually this ovule is attached to the placenta with the help of this stalk-like structure which is known as funicle. The junction between this ovule and funicle is known as hilum. Here the major part of this ovule is nucellus, which is living parenchymatous having the reserve food material, right? Having the reserve food material. Clear, bache? Clear, bache? And this nucellus is covered with the coverings and we used to call it as integument. So here we have 2 integuments, the outer one, inner one, and these integuments after fertilization, they are the one which will form the seed coat. Clear? So this portion is your nucellus. Okay, so integuments are not covering the tip region of this ovule. At the tip, integuments are missing. We are calling it as micropylar end. And always opposite to micropylar end, we will be having the chalazal end. That's what you need to remember. We will be having the chalazal end. Now, here, The ovule that we discuss in your syllabus is anatropous, the inverted ovule. Now in this nucellus, when we see this nucellus, see here in the hypodermal region means, see, if this is the epidermis, just below epidermis, hypodermis is there. So in a hypodermal region, you have one cell, you are calling it as archisporial cell. Now if I have to talk about the ploidy, even bache, your nucellus is diploid, even your integuments, because there is no meiotic division yet, so it is also diploid, right? Your megaspore mother cell, it is also diploid, but when it comes to your megaspore, it is haploid, right? Your female gametophyte, that is your embryo sac, overall it is also haploid. Clear bache? It is also haploid. So here this archisporial cell is dividing. This archisporial cell will form primary sporogenous cell and primary parietal cell. So this sporogenous cell will form megaspore mother cell. This sporogenous cell will form megaspore mother cell. This megaspore mother cell will undergo meiosis 1. You will get 2 haploid megaspores. And then you will undergo meiosis 2. Right? These megaspores will undergo meiosis 2. You will get 4 haploid megaspores. So out of these 4 haploid megaspores, only 1 will remain functional. The 3 will get degenerated. Clear bache? Clear? So one functional megaspore, it will develop into the embryo sac, which is the female gametophyte. Clear? Which is the female gametophyte. So such type of development where single spore is growing and forming female gametophyte is known as monosporic type of development. Clear bache? It is known as monosporic type of development. And here this megaspore, which is haploid. What is the ploidy of this megaspore? It is haploid. It will undergo 3 mitotic division. Right? So why 3 mitotic division? Because see, that's how it is going to grow. Because here we are going to see the development of the polygonum type of embryo sac. Right? So when it is the polygonum type of embryo sac, that's how it grows. So this functional megaspore will undergo 3 mitotic division. Right? It is also a previous year question. This functional megaspore, it will undergo 3 mitotic division. So 3 mitotic division means how many nuclei will form? How many nuclei will form? Right? There will be 8 nuclei. So this is actually the embryo sac which is developing here in nucellus bache. And this embryo sac, because here we will see the formation of female gamete. So that's why it is also known as female gametophyte. It is known as female gametophyte. Are you getting it? Are you getting it? That's how it grows. Clear? So now let's talk about this female gametophyte only. Okay, let's talk about this embryo sac. Done. So where is it growing? It is growing within the nucellus. Now after it, what will happen, bache? What is going to happen here? Now see, these cells will get arranged. Right? So initially only nuclear division was there. Now 3 nuclei will come to this near to this micropylar end side, 3 will come near to this chalazial end side, and 2 will move here in the center. 2 will move here in the center. Now what is going to happen, bache? These nuclei, right, they will get the walls, right? They will get covered with the coverings, right? They will have these cell wall around, right, bache? Right? Initially the mitotic divisions are just nuclear. Right? They are not followed by the cell wall formation. They are not followed by the cell wall formation. But now after the 8 nucleated stage, cell walls are laid down. Clear bache? Clear bache? So after 8 nucleated stage, right, cell walls are laid down. And when these cell walls are laid down, so obviously now there is the formation of a typical female gametophyte. Clear? See this. See this. Initially only nuclear divisions are there. This is the nuclei only. This is the nuclei only. 8 nucleated stage it is. 8 nucleated stage it is. Clear? Clear? Cells are not formed. But now after it, the cell wall will also start forming. So as I said, 3, right? As I said, bache, 3 cells will move towards this end. 3 nuclei will move towards this end. 3 nuclei will move towards this end, and 2 nuclei, they will move in the center. Right? They will come in the center. So now see, the development is ultimately there in the nucellus, yes or no? This development, students, ultimately it is there in the nucellus, yes or no? Yes or no? Ultimately this megaspore is developing within the nucellus, yes or no? Tell me. Imagine it like this, na? Right? Imagine it like this. Ultimately, this female gametophyte, the embryo sac, is developing within the nucellus. In the surrounding here, what do you have? You have the nucellus, and this embryo sac is taking nourishment from the nucellus itself. Do you know that? It is taking its nourishment from the nucellus itself. Now, can I say that when I look at this particular structure, can I say that just a minute? Can I say that near to the micropylar end you have 3 cells, near to the chalazal end also you have 3 cells? Can I say so? Near to the micropylar end also you have 3 cells. Near to the chalazal end also you have 3 cells. So now bache, these 3 cells which are near to the micropylar end, this central cell is your egg cell, right, which is haploid, and here, right, in its surrounding, these 2 cells are known as synergids, and they are also haploid. Just a minute, guys. Just a minute. Okay, they are also haploid. Okay. Is it clear? Yes. So near to the micropylar end, here also you are having 3 cells, right? Here also you are having 3 cells. So these 3 cells are actually forming the egg apparatus, right? They are forming the egg apparatus. So this egg apparatus is having 1 egg and 2 synergids. Clear, bache? This egg apparatus is having 1 cell and 2 synergids. Clear? 1 cell and 2 synergids. Done? So this egg apparatus is formed near the chalazal end, right? So it is present near to the micropylar end. Now when it is the chalazal end, so here you will see 3 cells. These are the antipodal cells. So their function is unknown actually. Okay, their function is unknown actually. Their function is not so clear. So antipodal cells are present near to chalazal end. Egg apparatus is present near to the micropylar end. And here in the center, bache, these 2 nuclei, they are known as polar nuclei. What is it? It is the polar nuclei. So here you will see N plus N arrangement. Right? What will you see here? You will see N plus N arrangement, means 2 haploid cells are there. And students, actually this complete portion, this portion, right? This portion, this is your central cell, right? This is your central cell, the largest cell of embryo sac, right? This is your central cell, the largest cell of embryo sac. Are you getting it? It is the largest cell of embryo sac. Are you getting it? See this one. So central cell is having 2 nuclei, 2 polar nuclei. N plus N condition is there. Which condition is there? N plus N condition is there. Clear, bache? Which condition is there? N plus N condition is there. So that's what I'm going to mention here. See, so in a typical embryo sac, also known as female gametophyte. So that's how you can revise. So near to micropylar end, you have egg apparatus, which includes 1 egg cell and 2 synergids, right? 2 synergids. So synergids are also haploid. So 2 synergid means again they are also haploid. So near to near to chalazal end, you have antipodal cells. They are also haploid. So you have 3 antipodal cells which are also haploid. So large central cell is also there, right? The largest cell of this egg apparatus, it is n plus n. It is n plus n. So if I have to count the number of cells, so 3 cells here, 3 cells here, and 1 large central cells. 1 large central cell. So in totality, how many cells are there? In totality, how many cells are there? 7 cells. So can I say that it is 7 celled? 1, 2, 3, 4, 5, 6. And this one is the large central cell. Okay, this one is the large central cell. See, this complete part, this complete cell is the central cell here. Okay, this complete cell is the central cell here. So central cell is having 2 haploid nuclei. Clear? Central cell is having 2 haploid nuclei. So this portion is what? It is the central cell having 2 haploid nuclei. Clear, bache? Clear, bache? So it's 7 celled but 8 nucleated. It is 7 celled but 8 nucleated. Okay? What is it? It is 7 celled and 8 nucleated. Done? Done? So it's the most common type of embryo sac. It is the polygonum type. Okay, it's the polygonum type. Clear? Clear? So it is developing here in this nucellus. So that's what you need to remember. And yes, it is very important. Okay, so let's start with the diagrams here. So you know that it is the pistil, right? This B is off. This B is off. Papaver. And this one is mycelia. Am I right? This one is mycelia. Am I right? Yes. And this is your ovule. So just look at the diagram here, bache. This junction is, see, hilum, micropylar end, chalazal end. The embryo sac is developing here. So here you can mention the ploidy as well. Nucellus diploid, integuments diploid, right? Embryo sac will be haploid. Rest everything is diploid. Okay. So look at this. So can you see that? Just in this nucellus, just below this epidermis here, this cell, this is your megaspore mother cell, again diploid. So it will divide meiotically, right? So it will form 2 megaspores, and because 2 megaspores are there, that's why we are using the word dyad. So when it is megaspore, it is haploid, but when it is megaspore mother cell, it is diploid. Clear, bache? Then after it, you will get 4 megaspores. Done, bache? Done. So in one functional megaspore, here you guys can see 1 mitotic division, then second, then third. So total it will be 8 nucleated. So after these 8 nuclei, what will you get? After these 8 nuclei, what will you get? The cell wall formation will be there. So here you can see that near to the micropylar end, there will be the egg apparatus, egg plus synergid, and near to this chalazal end, you will see antipodal cell, this large central cell having 2 polar nuclei. So here you guys can see, right? So why is it like this? Because you know that we have anatropous ovule in our syllabus, so it is inverted. That's why micropylar end is towards this side. Okay, the micropylar end is towards this side. Is that clear? Is that clear? And bache, when you look at these synergids now, these synergids are having filiform apparatus. We will see some projections here in the synergids, right? We are going to see some projections here in the synergids. We will call it as filiform apparatus, and this filiform apparatus is very important because it will, right, by chemotaxis, it is going to attract the pollen tube during pollination. When pollen grain will fall on stigma, pollen tube will form. So these filiform apparatus present in the synergid here, it will direct that pollen tube towards the egg, towards the female gamete. Is that clear? Towards the egg, towards the female gamete. Is that clear? So this portion is very important. If there is any doubt from this part, do let me know. Yes, if there is any doubt from this part, do let me know. Done, bache? Sure? Sure. So that's your female gametophyte. Clear? So just read this part from the NCERT, bache. We have covered each and everything. See, let me talk about the highlighted portion here. 2 nucleated, 4 nucleated, 8 nucleated, female gametophyte, right? So I hope you know the meaning of these terms. Clear, bache? I hope you know the meaning of these terms, right? So next topic is pollination that we need to start. But as of now, if there is any doubt, do let me know. As of now, if there is any doubt, do let me know. Do you have any doubt? Yes? Do you have any doubt? The next part that we need to start is your pollination. Okay, it is your pollination. So first of all, let's talk about the pollination, and then we will be discussing the outbreeding devices. No doubt? Okay. So you know that, bache, we are talking about the angiosperms, right? Right? And this portion we are going to cover from the NCERT only, okay? But let me explain you the meaning here. You know that here we are talking about the flowering plants. We are talking about the angiosperms here. What are we talking here? We are talking about the flowering plants here. We are talking about the angiosperms here, right? So you know that pollen grains are not motile. Clear? And that's your homework. You will tell me the answer in the comment section that what do you understand by the winged pollen grains? Fine, that's your homework. You have to tell me what is the meaning of the winged pollen grains. Are you getting it? What is the meaning of the winged pollen grains? Clear? And where are you going to find it? This is what you need to tell me. This is what you need to tell me. So we are talking about the pollination. What are we discussing? We are talking about the pollination. No doubt it is the transfer of pollen grains. It is the transfer of pollen grains from anther to stigma. From anther to stigma. That's what pollination is. Yes or no? That's what pollination is. So here the pollen grain, as I said, the pollen grain is not motile, right? We need some agents that can move that pollen grain from that anther to the stigma, right? So you know one thing, right? When I was teaching you the structure of this stamen here, you know that this part is the anther, you know that this part is the filament, and both of them together they are making what? They are making the stigma, right? They are making the stamen, right? Stamen, focus here. So anther plus filament is together making what? It is making the stamen. So you know that in the anther there will be the, right? You remember that stomium part? Do you remember that stomium part? Right, that dehiscence will occur, that longitudinal grooves are present in that thecal region. So the dehiscence will occur, right? I have in detail, I have discussed that mechanism. So the anther, so these pollen grains will be released. Now they need to fall on the compatible stigma. So obviously we need agents. So we will talk about that agents too, like we will talk about the wind as well, right? The pollination with the help of wind is also there, known as anemophily, right? The pollination with the help of water is also there, known as hydrophily. And sometimes, you know, animals are involved, right? Then we call it as zoophily. Especially your insects are involved, we call it as entomophily. If birds are involved, we used to call it as ornithophily. So that is what we need to discuss here. But before that, what we need to understand is that pollination is transfer of pollen grains from anther to stigma, and this can be of different type. Like, see, you know about these two words, I'm pretty sure about it, that one is the self-pollination and another is the cross-pollination. In the case of animals also, we talk about this, self-fertilization and the cross-fertilization. Self-fertilization and the cross-fertilization. Let me quote one example. I'll quote one example from the animals. Just let's talk about the earthworm. So if I talk about the earthworm, you know, farmer's friend, Ferretima posthuma, right? So a earthworm is hermaphrodite. What is the meaning of hermaphrodite, students? What is the meaning of hermaphrodite? What is the meaning of hermaphrodite? Remember that trick? It's BMH, bisexual monoecious hermaphrodite. Bisexual monoecious hermaphrodite. That is, it is having the male part as well as well as the female part. That a single organism is having the male part as well, female part as well. That is, it is having the gonad, it is having the testes as well, ovaries as well. But still earthworm is hermaphrodite, right? It is having the testes as well, it is having the ovaries as well, but still it prefers the cross fertilization. Do you know that? Like, this is the one worm, this is the another worm. It is going to pass its sperm to this worm and it will receive its egg like this. Still they prefer the cross fertilization. Do you know why? Because the cross fertilization promotes the variation, right? Cross fertilization, it promotes the variations, right? See, in the nature, you know that, like, even if I talk about the meiosis, in the meiosis, what is there? Crossing over is there. Crossing over will result in recombination. Recombination will result in variation, and variations are very important for the adaptations. Variations are very important for the survival. Same is the case here. So even in the case of flowers, we will also see self-pollination. So what is the meaning of self-pollination here? Like, single flower, it is having both, right? That the same flower is having both. Same flower is having both. It is having the anther filament as well, it is having the carpels as well, right? So pollen grains from this anther will fall here on this stigma. So self-pollination, the single flower is involved, its male and female part is involved. When it is the cross-pollination, so we will talk about different flowers, right? We are going to talk about different flower, like from this flower, right? From this flower, the anthers from this particular compatible flower will right, the pollen grains from the anther of this particular flower will form on the different stigma. Cross pollination, where you are talking about two genetically different flowers. Here you are talking about genetically similar, or you can say that same flower. Self pollination and the cross pollination. Self pollination and the cross pollination. So self pollination, when the same flower it is involved, or you can say that genetically same flower is involved. What is there? Genetically same flower, where their genes are same. Okay, genes are same. And when it is the cross pollination, where two genetically different flowers of same species are involved, right? So why am I emphasizing it? Let me tell you why. See, there can be two scenarios. Like, let's say this is a tree. This is a tree having flowers. Okay, this is our plant having flowers, whatever you want to say. So this flower is a bisexual flower. This is a sign for the bisexual flower. Here the flowers are bisexual. So if flowers are bisexual, can we say that there are the chances of self-pollination? Yes or no? If flowers are bisexual, can I say that there will be the chances of self-pollination or not? Of course I can say that, right? Let's not consider the outbreeding devices as of now. Let's not talk about any other mechanism. Let's simply talk about this fact that here the flowers are bisexual, and obviously self-pollination is possible here. Each and every flower is what? It is bisexual. It is bisexual. Now I'll give you another example. Let's say this is another plant. Here also it is having the flower. Here also it is having the flower. Now here the flowers are unisexual. In this plant, I am saying the flowers are bisexual. What are they? They are bisexual. So all in all, even your plant is also bisexual, that is, it is also monoecious. It is also monoecious. Am I right? It is also monoecious. Am I right? Am I right? Now look at this particular plant. Here the flowers are unisexual. Like, let's say this is the male flower and this one is the female flower. This one is the female flower. This one is the male flower. This one is the female flower. Clear? So plant is the same. This plant is having unisexual flower. Can I say that? This plant is having male flower as well, female flower as well. It is having the staminate as well. It is having the pistillate as well. Overall, if I talk about this plant, can I say that this plant is monoecious? Yes or no? Can I say that this plant is monoecious? Yes or no? Yes or no? Can I say that this plant is monoecious? Yes or no? Monoecious means bisexual. Bisexual. Yes, I can say so. Why? Because this plant is having the male flower as well as female flower, male flower as well as female flower, male flower as well as female flower. So definitely this plant is monoecious. Definitely this plant is monoecious. Are you getting my point? This plant is monoecious. But if I talk about the flowers here, flowers are unisexual. Flowers are unisexual here, right? Male and female flower is different here, right? But male and female flowers are present on the same, male and female flowers are present on the same plant. So overall plant is monoecious. So we see such example in the case of castor. In the case of maize, you will see these examples. In the case of castor and maize, right? In the case of castor and maize. So actually what's the scenario? Let me see, we have two plants. One plant is having bisexual flowers, right? One plant is having bisexual flowers. Another plant is having unisexual flowers. That's what I'm trying to say. That's what I'm trying to say. This plant is having bisexual flowers. This plant is having unisexual flowers. But again, when you look at these plants, here you will see male, female part both is present. Here, no doubt, male part is different, female part is different. Male part is different, female flower is different, but ultimately it is present on the same plant. These are the two cases that we have, okay? But both the plants are monoecious. So such type of example you will see in the case of castor and you will see in the case of maize. Such example you will see in the case of castor, you will see in the maize. But bache, here, even if you consider this flower, even if you consider these flowers, so don't you think that all the flowers which are present on this plant, they are genetically same? They are genetically same, can I say so? They are genetically same. Yes or no? Can I say that they are genetically same? And even if I talk about these flowers, although it's a male flower, it's a female flower, it's a male flower, it's a female flower, it's a male flower, it's a female flower, but they are present on same plant. So can I say that they are also genetically same? Can I say that they are also genetically same? Like let's say here, let's take the example of humans, right? Two siblings are there, right? So they are siblings, they have the same parents, they are living in a same family. Here also you are talking about two siblings, right? Here also you are talking about two siblings, right? So one is living in a foreign country and one is living here in India. But that doesn't mean that their, that their genes are not same. Of course, their genes are same. है ना? Let's say here you are taking the examples of the siblings. Siblings means they have the same parents, they are living in a same family. So obviously they are genetically same. Here also you are taking the example of the siblings. But right, one is living in a foreign country, let's say in Australia, one is living here in India. But that doesn't mean they are not genetically same. They are genetically same. They are genetically same. So here also we are talking about two types of flowers. Flowers are monoecious. We are talking about two types of plants. Plants are monoecious. They are bisexual, right? But here in one example, in case one, I am saying that flowers are bisexual, right? Here I am saying the flowers are hermaphrodite. But here I am saying that flowers are unisexual. They are dioecious. But male and female flower is present on the same plant. Right? So such example is castor and maize, where your plant is monoecious, but flowers are dioecious. Flowers are dioecious. Flowers are unisexual, but male and female flowers still present on the same plant. Right? It is still present on the same plant. Is that clear? Right? Is that clear? Right? But now you must be thinking that, ma'am, why are you explaining it? Why are you explaining it? What is the need? Actually, I need to relate it with the pollination. Thank you so much, Akhilesh. Okay, so in pollination, like I have used the word self-pollination. Self-pollination is in between the flowers which are genetically same. Like you have the same bisexual flower, right? The pollen grains here from this anther will fall on the stigma. So it is the self-pollination. It is the self-pollination, right? So when there is the bisexual flower, and the anther from the right? The pollen grain from the same flower, pollen grain from the same flower fall on the same stigma here, then it is known as autogamy. What is it, Suhashini? It is known as autogamy. But when you are taking this case, when you are, Vinod Bache, thank you, but when you are taking this case where, right, anther from this flower, right, the pollen grain from this particular anther will fall on the stigma of this female part. Okay, so this case is of geitonogamy. This case is of geitonogamy. So here flowers are genetically same, right? Ultimately flowers are genetically same. Yes or no? Yes or no? So both of them are examples of the self-pollination. This is autogamy, where you have bisexual flower. So from same flower, from the pollen grain from the same flower will fall on the same stigma. But here what is happening in geitonogamy? The flower is ultimately it is genetically same. Flowers are different, but genetically they are same because plant is same. Right? Plant is same. The example is castor and maize. And when there will be the different plants, like in the case of papaya, unisexual, male plant, female plant, right? So from male plant, the pollen grains will fall on female plant, such cases of cross pollination. That is your xenogamy. That is your xenogamy, because they are genetically different. I hope you have noticed. Then when we grow papaya, right, different types of, we grow many papaya plants together. Why? Because some of them are male, some of them are female plants. Okay, so now let's read it from NCERT. You will find it easy, trust me. So pollination, it is the preceding sections that we have to learn. Okay, after male and female gamete formation, this is what we need to understand now. Okay, so both the types of the male gamete and the female gamete in the case of plants, they are non-motile. Yes, ma'am, they are non-motile. So they have to be brought together for the fertilization, right? So that is your pollination, the mechanism to achieve this objective. It is the mechanism to achieve this objective. It is the mechanism to bring the male and the female gamete closer. So transfer of pollen grain from the anther to the stigma of the flower is the pollination. Clear, bache? Clear, bache? See, transfer of pollen grains to the stigma of a pistil is the pollination. So flowering plants, they have evolved an amazing array of adaptations to achieve this part, right? To achieve this part. So first of all, let's talk about the kinds of pollination. First of all, let's talk about the kind of pollination. So the very first kind is the autogamy. Auto means self, right? Auto means self. Right, bache? Right, bache? So pollination is achieved within the same flower. Flower is same, means flower is bisexual. Flower is same, means flower is bisexual. So transfer of pollen grains from the anther to the stigma of the same flower. What is happening here? Transfer of pollen grains from the anther to the stigma of the same flower, bache. So normal flower which opens and exposes the anther and the stigma, they will show complete autogamy. But complete autogamy is rare. So ultimately, what is autogamy? It is the self-pollination where anther of the same, right, pollen grain from same anther falls on the stigma of the same flower. So it is self-pollination. But let me tell you, and here your stigma needs to be exposed, your anthers needs to be exposed. But complete autogamy is rare. We will talk about it. Clear, bache? We will talk about it. And here what we need to observe, that bache, there should be the synchrony. Now what is the meaning of synchrony? See, let's say you have fight with your friend, right, or with your parents, whosoever is there. So obviously if you are going and telling them sorry, even they should be in receptive mode. Like if, let's say you made a mistake, you're coming to me, you're saying sorry to me. So if I am in that mood that okay, fine, it's okay, it happens, leave it, right, then only that patch up thing will occur, hai na? Otherwise though it will be of no use, right? If I'm angry, if I'm crossed with you all, even if you are saying sorry, it is not going to make sense. I need to be in receptive mode as well. Even I need to be that understanding. That's the point. Same is the case here. right? Same is the case here. If pollen grains are coming from this anther, even your stigma needs to be in receptive mode. Even stigma needs to be sexually mature, that okay, if pollen grain of same species will fall here, I will allow it to form the pollen tube. I will allow the pollination and the fertilization to occur. Right. So for the self-pollination, the synchrony is very important. Right. It should not be like this, that when pollen grains are released from this anther, the stigma is not in receptive mode. Then again, the fertilization will not occur. And stigma is in receptive mode, but anther is not mature, then also it cannot occur. So there should be the synchrony. There should be the synchrony. Clear bache? Clear bache? And anther, and see here, the anther and the stigma should lie close to each other, then only the self-pollination can occur. Here actually we will discuss 3 examples. Right? I'll tell you these 3 examples, then we will move to geitonogamy and xenogamy, then we will end the class. Rest we will discuss in the next class. Okay. So there are 3 examples. This is what you need to remember. Here 3, see, say VOC. What is it? It's VOC. Everyone, it's VOC. Viola oxalis. And do check their images. And C is your Commelina. Fine, C is your Commelina. So it's VOC, Viola oxalis commelina. It's VOC, Viola oxalis commelina. It's VOC, Viola oxalis commelina. What is it? It's VOC, Viola oxalis and commelina. Clear, bache? Now these are the 3 examples where what will you see? You will see flowers. 2 type of flowers are there. Right? Open flower will be there. Closed flower will be there. Open flower will be there. Closed flower will be there. Closed, clistogamous. See, even spellings are same. है ना? Closed, CL. Clistogamous. Open is chasmogamous. Open is chasmogamous. Right? Here we are discussing the orthogamy, and then we are discussing 3 examples. We are discussing the examples of VOC. What is VOC? Viola oxalis commelina. Everyone just repeat it here in the chat section. Viola oxalis commelina. Viola oxalis commelina. Viola oxalis commelina. That's what we are discussing. So here flowers are of 2 types, open and the closed. What is the meaning of open part? Where your anther is exposed, where your stigma is exposed. They are exposed, they are open. And when it is a closed flower, let's say like this, that stigma, that anther, they are not exposed, they are present like this, closed. In a closed system they are present. Now see, if it is open enough, right, the stigma is open enough, right, this anther is open, it is exposed, the stigma is exposed. So do you think that it's mandatory? Do you think that it's mandatory that always this stigma will receive pollen grains from this anther only? What do you say? It is open, it is exposed. It is open, it is exposed. So obviously this stigma can also, like let's say with the help of wind, pollen grains can, my pollen grains can come from another flower also. Yes or no? Yes or no? So can I not say that here complete autogamy is not there? Here cross-pollination can also occur. It is not just the self-pollination that can occur here. The cross-pollination can also occur. The anther from, the pollen grain from some another flower can also fall on the stigma. Right? Right? Yes or no? But when it is totally closed, when it is a closed flower, when it is a clistogamous flower, when it is a closed flower. Now in this case, what's the scenario? Right? In this case, what is the scenario, bache? What is the scenario here in this case? Because it is totally closed, so obviously these pollen grains will definitely fall on this stigma only. So 100% self-pollination will be there. Right? Assured seed set will form and 100% self-pollination is there. So 100% autogamy will occur here. So these examples are very important. Even in bache CBSE exams, right? Even in CBSE exams, you used to get question from this part. Right? So here in clistogamous flowers, you will see that autogamy will be there. Only here the chances of cross-pollination are also there, right? Chances for the cross-pollination are also there. So see these examples. Like see some common plants, your viola, common pansy, oxalis and commelina. They are having 2 type of flowers, chasmogamous, which are similar to flowers of other species, right? Having exposed the anther and stigma. Clistogamous, which do not open at all. Right? See chasmogamous clistogamas, right? So which do not open at all. So in such flowers, anther and stigma lie close to each other. So when anther dies, in the flower bud, pollen grains come in contact with stigma. So in clistogamous flowers, invariably autogamous it is. No chance of cross-pollination will be there. Assured seed set, even in the absence of pollinators, will be there. Is that clear? Is that clear? But again, the clistogamy is having disadvantage. You know that. What is it? What is it? Inbreeding only. It can result in inbreeding depression, right? Because same genes are there. So it will not result in variations, and you know that variations are important. Next example is of gitonogamy. Transfer of pollen grains from the anther to the stigma of another flower, but the plant is same. Guys, read it carefully. Read it carefully. Here also there is a transfer of pollen grains from the anther to the stigma of another flower of the same plant, right? Right, so transfer of pollen grains from the anther to the stigma of another flower of the same plant, right? Of the same plant. So here, two flowers are different. Like I have given you the example of castor and maize, where plant is going to be same, flowers are different. Plants are going to be same. Plants are monoecious, but their flowers are dioecious. Clear, bache? Clear, bache? So gitonogamy is functionally cross-pollination. What is the meaning of functionally cross-pollination? Let me explain it to you here. So if I need to transfer the pollen grain of this flower to this stigma here, so obviously I need pollinators. I need pollinators here. So that's why I'm saying that it is no doubt, it's kind of cross-pollination, because I need the involvement of pollinators here. But ultimately, because plant is same, so genes are same, right? Genetically it is similar. So it is genetically, it is the self-pollination. Clear, bache? Clear, bache? So genetically it is similar to autogamy. Clear? Clear? Next is the xenogamy, the true cross-pollination. So transfer of pollen grains from anther to the stigma of a different plant, but yes, the species needs to be same. So genetically different plants will be considered here, like your papaya. Clear? Like your papaya. Now, bache, I have a class, right? After this a YouTube class, I have a class in my plus batch, basically in Avengers 2.0 batch, right? So today we are going to continue with the digestion and absorption there, and hopefully I think it's 5th. So on 8th of September, we'll be starting breathing and exchange of gases, right? So we have just started the human physiology, okay, in Avengers 2.0. So that's a request, bache, if you are seriously interested in joining a batch, because still, if you will even miss the month of September, or even if you will delay for even a single day or for 2 days, obviously there will be a lot of backlogs then. So join the batches. Okay, if you are serious for this NEET examination, and if you have decided that you are going to prepare it, right, sincerely, then this batch is for you, because in very less amount you are getting such quality content. And yes, in offline coachings you used to pay in lakhs for getting this level of content. Okay, so join the batch as soon as possible, and from these agents of pollination we will continue tomorrow, right? So please make the notes, please revise it accordingly. Okay, please make the notes, please revise it accordingly. Do check this particular video as well, this strategy video, right? It is an eye-opening video for all of you. And what you have to do, see, you have to click on this link if you want to be the part of this particular batch. You have to click on this link. It loading takes some time, but all the details will come here. So what you have to do, you have to add your number, you have to add your email ID, right? Then after paying the fees, you will be the part of this batch. So as I said, bache, as I said, 70% of the content is still live. Okay, right? It is just the 30% that will be in the recorded form. See, human physiology we are discussing. After that, I will start your class 12 syllabus, the genetics, the biotech, the ecology. So still you have time. So enroll in this batch as soon as possible. Just add your number, or you can continue with email. You can see the Avengers coupon code is already applied, and at a price of 4999 you will get this batch. Okay, and plus mock test as well, plus doubt sessions as well, plus revision as well, right? And after few days we will end the enrollments for this particular batch, because we know now later on it will be very difficult for you to manage. It will be of no use to be the part of any batch then, right? If you have prepared the syllabus, then okay, crash course is fine. Otherwise this detailed batch is not for you. Okay, so join it as soon as possible. And now you know what you have to do. I'm going to end the class, right? It will hardly take you a few seconds, and it's a Teachers' Day, of course. So this is what I'm expecting from you people. Just put a comment in the comment section, right? That how much you like this class. Okay, so thank you so much. See you all today at 5 p.m. for that roundtable discussion, right? Let's chit chat. We will guide you as well. And if you have any doubts, just be the part of that particular class, right? Which is at 5 p.m. today. Okay, so thank you so much. See you all tomorrow at 10 a.m.